SANDHYA as Associate Professor of Emmanuel Rasta College of Pharmacy. Today we are going to discuss about the topic of the isolation and the preservation of culture of microorganism. What do you mean by isolation and preservation of cultural organism and microorganism? Introduction. Microorganisms are generally found in the natural air, soil and water as mixed populations. Even the diseased part of plants and animals contain a greater number of microorganisms. Study the specific role played by a specific microorganism in events environment. One most isolated the same in the pure culture. Pure is mixed culture. Pure means originated from first bacteria strain. All colonies look the same. Mixed originated from many bacteria strains. The colonies have different size and shape. There are several methods of isolating the pure culture of bacteria like streak plate method and pure plate method and split plate method and serial dilutions. The important pure cultures are important in microbiology for the following reasons. Once purified, the isolated species can then be cultivated with the knowledge that only the described microorganism is beginning growth. A pure culture can be currently identified for accurately studying and testing and diagnosis in a clinical environment. Testing experimental with a pure culture ensures that the same result can be achieved regardless of show. Many times the test is replicated. Common methods of isolation of pure culture. The process of screening a pure culture by spreading one type of microbes from a mixture is called isolation. Common methods of isolation: streak plate method, pour plate method, serial dilution techniques, and the spread plate method. Then first one, special method of isolation of pure culture: single cell isolation method, capillary peptide method, micromanipulator method, enteral cultural media. Splitting is a technique used to isolate a pure strain from a single species of microorganism, often bacteria. Samples can then be taken from the resulting colonies and microbiological culture can be grown on a new plate so that the organism can be identified, studied or tested. Then the inoculum is a snapshot of the vanilla. Loop is retraced and parallel lines drawn from the wall. Loop is retraced and parallel lines are down from primary stroke. Loop is retraced and parallel lines are down from secondary stroke. Loop is restricted and zigzag lines are down from tertiary stroke. Appearance of isolated colonies towards the trapping end. Then a diagrammatic result showing the streak plate method, streak cultures. Streak culture various methods of streaking. Various methods of the streaking. Pure plate method. The bacterial culture and liquid agar medium are mixed together. After mixing the medium containing the culture produced into sterilized petri dishes allowed solidifying and then incubated. After incubation, colonies appearance on the surface. The pure plate method 1 ml origin and uniform dilutions 1.10 1.100 1.1000 1.10,000 1.100,000 0.1 ml plating. This is pure plate method. Disadvantages of pure plate. These microorganisms are trapped beneath the surface of medium when it is solidified. Hence, surface as well as subsurface colonies are developed, and it is very difficult to isolate and count the subsurface colonies. This method is thorough, time consuming and requires skill. These Microorganisms are subjected to hot stroke because liquid medium is maintained at 45 degrees Celsius temperature, unsuitable for isolations of psychochlorophylla bacteria. This is the disadvantage of the chloride method. These microorganisms are trapped beneath the surface of medium when it is solidifies, and surface of the subsurface colonies are developed, and it's very difficult to isolate and count the subsurface colonies. This method is relatively time consuming and requires space. The microorganisms are subjected to hot stroke because liquid medium is maintained at 45 degrees Celsius temperature, unsuitable for isolation of physiocytotic bacteria. Procedure for street and pure plate method. Spread plate method. Sample is peptide onto surface of agar plate 0.1 m or less. Sample is spread evenly over surface of agar using a stirling glass cutter. Inoculation. Topical spread plate result surface colonies. Pure plate method. Sample is peptide into a sterile plate. Sterile medium is added and mixed well with inoculum. Incubulations. Surface colonies. Typical pure plate method. Result. Procedure for the spread and pure plate method. Spread plate method and pure plate method. Spherical dilution. This method is commonly used to obtain pure cultures of this microorganism that have not been successfully cultivated on solid media and grow only in liquid media. A microorganism that primitive in a mixed culture can be isolated in a pure form by a series of dilutions. The inoculum is subjected to serial dilutions in a sterilized liquid medium, and a large number of tubes of sterilized liquid medium are incubated with liquid of each successive dilutions. Colony of bacteria. Sample to be conducted. If we take out 1 ml of this medium and mix it with 9 ml of fresh sterilized liquid medium, we would then have 100 microorganisms in 10 ml of 10 microorganisms. If we added 1 ml of the suspension to another 9 ml to fresh sterilized liquid medium, each 1 ml would now contain a single microorganism. If this tube shows microbial growth, there is a very high probability that this growth has resulted from the introduction of a single microorganism in the media and represented the pure culture of that organism. Spread plate method. This is the best method to isolate the pure colonies. In these techniques, the culture is not mixed with the agar medium. Instead, it is mixed with the normal solvents and sterilized diluted. 0.1 ml of sample taken from diluted mixture, which is placed on the surface of the agar plate and spread evenly over the surface by using L-shaped glass rod called a spreader. Incubulated the plate. After colonies are observed on the agar surface. Colonies are observed on the agar surface. Taken from diluted mixture, which is placed on the surface of the agar plate and spread evenly over the surface by using L-shaped glass rod. Then advantages of the spread plate method. It is a simple method. In this method, only surface colonies are formed. Microorganisms are not exposed to higher temperature. It is a simple method. In this method, only surface colonies are formed. Microorganisms are not exposed to the high temperature. This is the spread plate method. Spread plate method.